Plant viruses represent a fascinating and diverse group of pathogens that have evolved specifically to infect plant hosts. This diverse group includes approximately 1,300 distinct species, organized across 90 genera and 25 families. This classification system organizes plant viruses into a clear hierarchy, from broad families down to specific species. Plant viruses share fundamental characteristics with all viruses, but have evolved unique features specifically for infecting plant hosts. Plant viruses have tremendous significance in both agricultural systems and natural ecosystems, causing widespread disease and economic impact. As we delve deeper into plant virology, let's zoom into the microscopic world to explore their structure and composition. Plant viruses have a simple yet elegant structure consisting of two main components. At the center is the nucleic acid, which contains the virus's genetic material. Surrounding this genetic core is a protective protein coat called the capsid. The capsid assembles around the nucleic acid to form a complete virus particle. Plant viruses come in three main structural forms. Rod-shaped viruses are cylindrical with a rigid structure. Filamentous viruses are long and flexible like threads. Icosahedral viruses have a spherical shape with geometric symmetry. The genetic material of plant viruses can be either RNA or DNA. RNA viruses are by far the most common, with single-stranded RNA being the predominant form. DNA viruses are much less common in plants compared to animals. Approximately 95% of all plant viruses contain RNA as their genetic material. Plant viruses differ significantly from human viruses in their genetic composition. While plant viruses are predominantly RNA-based with limited diversity, human viruses show much greater diversity in both genetic material and structural complexity. Understanding virus structure is essential for studying how these pathogens enter and infect plant cells. Plant viruses face a unique challenge when trying to infect plant cells. Unlike animal cells, plant cells are surrounded by a rigid cell wall that acts as a protective barrier. Plant viruses cannot actively penetrate this rigid barrier on their own. This contrasts with animal cells, which have flexible membranes that viruses can bind to and penetrate directly. Plant viruses require wounds or damage to the cell wall to gain entry. Insects can create wounds through feeding, creating entry points for viruses. Agricultural tools and equipment can also damage plant tissues, creating entry wounds. Environmental factors like wind, hail, or physical abrasion can also compromise the cell wall. Once wounds are present, viruses can successfully enter plant cells and begin infection. Understanding these entry mechanisms is crucial for developing effective plant disease management strategies. Plant viruses rely on various methods to spread between host plants, with biological vectors being the most common transmission route. The primary biological vectors for plant virus transmission are insects, particularly aphids, whiteflies, and leafhoppers. Unlike in animal hosts, viruses typically do not replicate within insect vectors. Instead, insects serve as passive carriers, transporting viruses from infected plants to healthy ones. When an insect feeds on an infected plant, it picks up virus particles. The insect then carries these viruses to the next plant it feeds on. Plant viruses can also spread through other methods, including seed transmission, vegetative propagation, and mechanical transmission. Seed transmission occurs when viruses are carried within infected seeds to the next generation of plants. Vegetative propagation spreads viruses through grafting, cutting, or division of infected plant materials. Mechanical transmission happens when virus-containing sap from infected plants enters wounds in healthy plants. The efficiency of virus transmission varies significantly between methods, with biological vectors and vegetative propagation being among the most effective. Understanding virus transmission is crucial for developing effective management strategies and preventing the spread of plant diseases. Now we zoom in to see how plant viruses move from cell to cell at the microscopic level. Plant cells are surrounded by thick cell walls and are connected by specialized channels called plasmodesmata. When plant viruses infect a cell, they need to move to neighboring cells to spread the infection. Viruses encode special movement proteins that modify the plasmodesmata channels to make them wider. These movement proteins increase the size exclusion limit of plasmodesmata, allowing viral nucleic acids or particles to pass through. The viral particles can now move through the modified channels into neighboring cells. This mechanism is unique to plant viruses. Let's compare this with how animal viruses spread. Plant viruses have evolved to use movement proteins and modify plasmodesmata for spreading between cells. In contrast, animal viruses typically spread by breaking cells apart or budding from cell membranes. This movement protein mechanism is a key adaptation that allows plant viruses to overcome the challenge of rigid cell walls. Understanding this cell-to-cell movement helps explain how plant viruses establish systemic infections. As plant viruses establish infection, they cause visible symptoms that develop progressively throughout the plant. Mosaic symptoms appear as irregular patches of different colors on leaves, creating a mottled appearance. Chlorosis is the yellowing of plant tissue due to loss of chlorophyll, progressing from mild to severe. Viral infections often cause stunting, where infected plants show significantly reduced growth compared to healthy ones. Necrosis represents the most severe symptom, where plant cells die and tissue turns brown or black. Symptoms develop progressively as viruses move through the plant's transport system, starting from infection sites and spreading to new tissues. Understanding these symptoms helps in early detection and management of viral diseases in plants. Now we explore how viruses replicate within plant cells, hijacking the host's cellular machinery. Viruses enter plant cells and begin the process of hijacking cellular machinery for their own replication. The virus redirects the cell's ribosomes from producing normal plant proteins to manufacturing viral proteins instead. The virus also hijacks the host cell's DNA and RNA replication machinery to copy its own genetic material. Multiple copies of the viral genome are produced, using the host's energy and resources. This replication process diverts cellular resources away from normal plant functions, contributing to disease symptoms. Different plant viruses use different replication strategies depending on their genetic material, RNA or DNA. This viral replication process triggers the plant's natural defense mechanisms, which we'll explore next. Plants employ RNA silencing as their first line of defense against viral infections. When viral RNA enters the plant cell, it is detected by the plant's defense machinery. The plant produces small interfering RNAs called siRNAs that specifically target the viral RNA sequence. These siRNAs are loaded into the RISC complex, which acts like molecular scissors to cut and degrade the viral RNA. The viral RNA is then cut into small fragments and degraded, preventing viral replication. Plants also defend themselves using resistance proteins, or R proteins, that act like molecular sentries. These R proteins are designed to recognize specific viral components called elicitors. When an R protein recognizes a viral elicitor, it triggers an immediate defensive response. This recognition event sends defense signals throughout the plant cell, activating multiple protective pathways. Plants can develop systemic acquired resistance, a form of immunological memory. When a plant experiences viral infection in one area, it sends warning signals throughout the entire plant. These signals travel through the plant's vascular system, preparing distant tissues for potential future attacks. The uninfected parts of the plant enter an enhanced defense state, making them more resistant to subsequent viral infections. Plants have evolved a sophisticated defense arsenal combining immediate RNA silencing, protein-based recognition systems, and long-term acquired resistance. These layered defense mechanisms work together to protect plants from viral infections and limit their spread. Plant viruses cause devastating economic losses worldwide, with annual crop damage exceeding $60 billion globally. These viruses can reduce yields by 10 to 100% in infected crops, with major impacts on citrus, potato, and tomato production. Effective virus management requires multiple integrated approaches, starting with traditional strategies. These include using virus-free planting material, developing resistant varieties, controlling insect vectors, and implementing proper cultural practices. Modern biotechnology offers powerful new tools for virus management, including genetic engineering approaches. Early detection systems using advanced diagnostics help identify infections before symptoms appear, enabling rapid response. Successful virus management requires an integrated approach, combining traditional and modern strategies. Prevention remains the foundation, with resistant varieties and biotechnology providing additional layers of protection. Early detection and continuous monitoring are essential for long-term success. Through integrated management strategies, we can significantly reduce the economic impact of plant viruses and protect global food security for future generations.